In this lecture, we are going to explore a group of important biomolecules referred to as the nucleotides or nucleic acids, and we are going to look at both what they are and how they are biosynthesized in a cell. Now, nucleotides or nucleic acids contain three subparts in their structures. They are composed of a combination of a nitrogenous base, a sugar, and phosphoric acid. Now, nucleotides are sometimes also referred to as phosphorylated derivatives of nucleosides. In this regard, a nucleoside by definition is a combination of only a nitrogenous base plus sugar. Thus, the phosphorylated derivative of a nucleoside would now have three parts, and that would be called as a nucleotide. Now, as far as the sugar is concerned, the sugar which is present in these nucleosides or nucleotides is either ribose or deoxyribose. If you recall, ribose is a pentose, and here is the structure of ribose. Deoxyribose is the 2' deoxy derivative of ribose. So this pentose is an integral part of both nucleosides as well as nucleotides. Now, as far as the nitrogen base part is considered, the nitrogenous base can chemically be of two types. Either it could be a purine, and this is the structure of a typical purine entity, or it could be a pyrimidine. Note that both purine and pyrimidine are nitrogen containing heterocyclic compounds. A pyrimidine is a ring form containing two nitrogen atoms, while purine is bicyclic and contains 4 nitrogen atoms. Before we explore the structures of the different purine bases and pyrimidine bases that are part of nucleosides and therefore later part of nucleotides, let us very quickly look at the important biological functions of this group of molecules. The nucleotides or nucleic acids serve some important biological roles. One is that they are integral part of DNA and RNA. In fact, DNA, which is deoxyribonucleic acid, or RNA, which is ribonucleic acid, are nothing but polymers of these nucleotides, and they play an important role in both storage of genetic information and the transfer of genetic information through RNA to proteins, that is, the reading of the genetic information. A second important role of nucleotides can be seen in the form of ATP being considered as the energy currency of the cell. Many biological enzyme reactions take place because of the energy provided by the hydrolysis of ATP to ADP and phosphate. Another biological role of these nucleic acids or nucleotides is that they are also second messengers in many of the information transfer pathways. For example, cyclic AMP and cyclic GMP are well-known second messengers which allow the signals due to binding of, say, ligands and hormones or receptors to be passed on to the inside of the cell.